Orchid Society. So anyone can go to orchidsanfrancisco.org and access the public site, but we also have a member-only portion. So if you have not logged in and created a password for the member-only portion of the site, and you're confused on how to do that, I'm happy to help. You can just ping me at johnrush@orchidsanfrancisco.org. That's my email.
So can you guys see this? This is our website, and I just logged in to my account, and then what happens is when you get in, you can actually see a member's homepage, and there's additional content on here, including the video archive links and the newsletters and etc. inside, as well as the usual stuff outside. And under more, you'll find all the other information. So
So Jeff, this is Sherry.
Yeah.
I've tried signing in several times, and that page comes up and says, you do not have access to these pages when I sign in. So
I'll help you out, Sherry.
thank you.
Yeah, if anyone has an issue, send a help desk ticket direct to our info@San Francisco.
I love it.
Yeah, we do answer these. So
I have a question. Where was the Orchid Digest link? Members meeting?
it is in the events section. Let's see.
I'll drop a link into the Zoom chat.
It's also on, if you go on Facebook and just search for Orchid Digest Friends, it's posted there as an event, and you can.
Facebook, what's that?
All right,
Okay.
without further ado.
Go to orchiddigest.org.
I
Yes,
couldn't
it's
find
also
it there either.
posted on orchiddigest.org.
All right, I'll look
I
some more.
texted you a link to your phone this afternoon. Check your text.
All right, thank you.
Okay.
Some of us are Luddites, you know.
So I've eaten up a lot of time.
Luddite is the right word, but okay.
And Lynn has graciously been preparing and running our show and tell for us. And I think she's doing a little skill session topic too tonight. So can you get off mute and share your PowerPoint?
Sure. Share.
Okay. Okay, let's see.
What do you see?
I see your screen, and it's not in presentation. It's got a preview mode. Let me see here. Can everyone see it well? Yep. All right, off we go.
Yeah, we need the preview controls back.
We need the preview controls back.
That's up to us. For some reason,
Is it?
let me figure out how to get back. Okay. Do this.
Ooh, pretty.
Okay, that's good.
If you're wondering how to make your slideshow show up, you have to start the slideshow, then share the large slide. You can't share PowerPoint. You have to share that first slide. So whichever of those is your first slide, share that one.
There, can you see that?
No, see, they're
There.
seeing,
Well,
we're on a two-screen system here. So, oh, here we go. I just needed the controls.
It's better. It's not
You
great.
share, just a second, we'll get it. All right. Don't want that, don't want that. There we go. We just have to choose the right screen to share. Now you should see a full screen that just shows you the PowerPoint, correct?
Yes, that looks good.
Yep.
Perfect. Let's see if it,
Hey.
tell me if the screen changes. Did it change?
Hey.
Yes, all right.
Yes.
Okay.
Yes,
All right.
yes.
Okay, thanks, sweetie. So SFOS asked that individuals submit up to 3 orchids for show and tell. And some folks have asked that I just do 1 or 2 orchids per page. So they're larger and they're easier to see. So I'm giving that a try tonight. It's great if you send a photo of the plant and a close-up of the flower. Those will share a page, as you'll see. And there's a little surprise at the end. Got a lot of great photos tonight. The first one is from Deborah Vels-Qualters. This is her Encyclia Cindy. It's a primary hybrid of Encyclia elata and Encyclia tempense, which is widespread in Florida. It forms a really beautiful, graceful, arching spray. And Deborah says it smells like lilac. So what's not to love about that? Next, from Jeff Harris, we have this Sobralia veitchii, a primary hybrid of Sobralia macrantha byzanthiluca. It's interesting, if you look at awards for Sobralias, many of them have never been awarded, which is probably because the flowers are only open for a day or 2 and then they're gone, so they never get to judging. But many of them do produce 3 or 4 sequential blooms on an inflorescence. And these are good outdoor plants in the Bay Area. They love strong air movement, so they love the bay breezes. Give them a try, Sobralias. Next, from Jeff Harris. Jeff says his Stelis emarginata has been in bloom for almost 6 months in his greenhouse. There are hundreds of 1 to 2 millimeter flowers and about 35 inflorescences, each with about 25 flowers. So this is a really robust plant. It has Jeff says it has a prominent but pleasing musk fragrance. This is a species from Mexico and Guatemala, and it grows between 3,000 and 7,200 feet. So again, it can take our cool Bay Area temperatures. Also from Jeff, this is his first bloom seedling of Encyclia Marii, another Mexican species. There's a lot of flower here for the size of the plant. And the lip is just spectacular with green veins at the base of the lip. It's interesting to see here how Jeff hangs his mounted plants, including those that are in pots, outdoors in his growing area. And from Jeff, this is a good story. Jeff recently got this Diplocolobium arachnoideum from Dave Hermeyer. It's a dendrobium relative. They're actually tiny basal pseudobulbs down there, and it's found only on Papua New Guinea. Jeff tells me he was thrilled to see a pink bloom, but then he was puzzled when the next flower came out white. Then it turned pink, and it closed up and it faded to red. As you can see in the photo on the left, the whole plant, he did some research and he learned that the flower life is measured in hours, not in days. A white flower typically opens in the morning and is gone by late afternoon, but it can flower many times a year. Interesting plant. Tom Pickford sends a couple of eye-popping reds. On the left, a mounted Cattleya Circle of Life, which is a cross of Cattleya culminant by coccinea. This was made by Frank Fordyce in 1998. It's been used a lot in breeding, hybridizing. And on the right, a complex hybrid, Rhyncholalia Cattleya Paradise Rose, which gets its striking color from coccinea. Excuse me, the smoke is getting to my throat. And Dowiana has influenced the long ruffled lip. Very beautiful. Lost my mouse, there we go. Also from Tom, we see 2 white dendrobiums. On the left is Dendrobium sanderi, a species from the Philippines. The throat is typically maroon on this species. So this is a naturally occurring variety called Lusanicum. And on the right, excuse me, on the right is a complex hybrid Dendrobium snowbells, a recent cross made by Wright Tokunaga of H&R. And it's one of the grandparents is Sanderi on the left. So the one, snowbells has beautiful round full petals. The flower, I believe, is about 4 inches wide. Susan Anderson shows a Dendrobium cumulatum. This is from India and Southeast Asia. It prefers intermediate to a warm environment. The flowers are about 2 inches with a beautiful pattern of rosy spots on the lip. And I believe that's fragrant as well. Susan also shows an Angraecum longiscot. This is a primary hybrid of Longicalcar and Scottianum, thus the name longiscot. It has creamy white flowers with a 9 or 10 inch light green spur, nectary. This is one of my favorites because I like to say the name. Podangus dactyloceras. Okay, everybody say Podangus. Do you love the word Podangus? So this is a cool to warm growing African species with a charming spray of flowers that are about 1/2 inch. They're translucent white with a crystalline texture, sparkly texture. And I think this compact little grower should be in everyone's collection. It's good from windowsills to greenhouses. Podangus. From Heidi Arno, we have a complex hybrid in the Untitled family, Violara Patricia McCulley. She says it is exciting when one of her windowsill orchids blooms. She bought this at POE or at Orchids in the Park, and it's been in her kitchen window for 7 or 8 years. She's bloomed it twice before. This is a nice big flower with really interesting and intricate markings. I guess all of that to attract a pollinator. And this is a plant I purchased from this Vanda tessellata. I purchased from Deborah Atwood of Napa Valley Orchids in 2015. And it had long roots, which soon reached the greenhouse floor. So you can see at the bottom of the left-hand picture, I wrap it up in an ever-growing loop with Velcro ties. It's a species from India and Burma. And I love the lattice-like pattern or tessellations, just like on a giraffe. You see the giraffe on the bottom with its tessellations on the petals with a violet lip. This grows on my water wall, so it gets watered every day and it likes very bright light. And Andy Easton gave me a small plant of Miltonia or Miltoniopsis bluana in 2015. He made this albacross of Miltonia roselii and vexillarius using orzolan, which influences or initiates polyploidy. So in 5 years, it's gone from first bloom seedling to this huge and floriferous plant with just amazing fragrance. It fills the greenhouse. It blooms a couple times a year, but it never is at its prime for judging night. So I'll keep hoping. And Cypripedium suavis is a small, warm growing native of Panama to Costa Rica. I grow this in a 6-inch basket because the spike needs to come out the side, just like it would on a Stanhopea. Carol Zotowski donated this plant to the CSNJC Speakers Day auction in 2019, and I was the lucky high bidder. This is the first time it's bloomed for me. And Yunor Peralta gave me a bare root division of his Cattleya maxima semi-alba striata in 2016. This is probably the lowland form of maxima, as the pseudobulbs and the leaves are relatively large. Again, you can see we grow roots really well in our greenhouse. This is the loveliest of the maximas in my collection, and it currently has 3 inflorescences, each with 8 flowers. It's native to Ecuador, Peru, and Colombia. We'll see some other forms of it later in the slideshow. Excuse me, I need a sip of wine. This will help my throat. Maybe I should gargle. Dave Hermeyer starts out with a longtime favorite, Oncidopsis Nellie Eisler. This is a complex hybrid with a family tree of a bunch of different Miltonias crossed with Oncidinae. And you can see it's a really well-established plant. Thank you for the ruler, Dave, to give us a sense of the size of the plant. It has deep scarlet petals and sepals that are shown off by that huge spotted striped lip. Lovely flower presentation too. They're all looking forward at us. And Dave got this from the free San Francisco Orchid Society December raffle table. It's always a good story. Dave also shows us this Masdevallia chaparensis from Bolivia, which grows at elevations from 8,000 to 9,000 feet. So this is clearly a good cool grower for the Bay Area also. The clonal name Rip Hoff was given to it by its former San Francisco Orchid Society member, Pui Chin, in 1988 when he got an 80-point AM and an 81-point CCM culture award on a plant that had 29 flowers and 9 buds. So I think there must be a good story behind the Rip Hoff clonal name. And my guess is that he expected it would receive a higher score. It has never received a higher AOS award. So Dave, it looks like your plant could do with visit to the judging table. This is beautiful. Dave also shows us a Brazilian species, Laelia crispa, form vinicolor with large, long-lasting, fragrant flowers. The sepals typically roll back like this. You see them rolling back so they almost look like narrow tubes. The lip is the main attraction here, with rich purple marks and veins and flaring and wavy margins. And Dave brings us this much coveted Phalaenopsis traderjoensis.
Traderjoensis.
There's always something special to be found at Trader Joe's. The flowers are nicely presented here, alternating along the inflorescence. Very nice. Traderjoensis. And Paula Shu shows us a lovely compact RLC or Potanara hybrid, California Queen by RLC Loud 9. And it is Loud 9, not Cloud 9. I looked it up. This is a Sunset Valley Orchids, Fred Clark cross. And Potanaras are also great windowsill plants because they stay compact like this. Roberta grows outdoors at her home in Costa Mesa, California, about 4 miles from the coast. So she says the climate is similar to the Bay Area, but a little warmer, with winter lows in the 40s. She grows this Vanda roblingiana outdoors in filtered sun. It's native to the Philippines, around 1,800 meters, so quite cold tolerant for a Vanda. And the chestnut color, I think, is really lovely, as is the bilobed ruffled lip. Roberta also shows us Lycastes schilleriana. She says she grows all of her Lycastes in a shady area on the north side of the house, outdoors. The flowers have a very heavy substance, and as they age, some of the red in here fades to green, changes from bronze to green. The species is native to Colombia at elevations of 1,200 to 1,800 meters. And Roberta shows us the South American species Catasetum sacatum. She grows this outdoors, excuse me, in filtered sun from April through October, when the nighttime temps are typically 55 degrees or higher. Then she moves it to her greenhouse for the winter to keep it perfectly dry and a bit warmer. The habitat ranges from 300 meters to 1,700 meters. So it's a fairly cold tolerant in the winter as long as it's kept dry. And as you know, when dormant, these Catasetinae don't take up much space because all the leaves are gone. And having outdoors when in active growth gives Roberta more space to let it spread out. That pendant inflorescence is nearly 2 feet long. And Roberta brings us this very unusual, I'm going to pause to say this name, Neoconia monophylla. Neoconia monophylla. It's named for the Belgian botanist Conio. So there are only 2 species in this genus. The monophylla is native to the Blue Mountains of Jamaica at elevations of 1,600 to 1,800 meters. It's a miniature plant, as you can see from the inset in the upper right-hand corner, with Roberta's hand in there. The flower is about 2 inches wide, and she grows this outdoors shady and damp. I haven't seen this before. This is really lovely. John McCallan shows us this exquisite Phragmipedium QF Halia. QF is Quintal Farms in Hawaii. Some of you may know them. They're particularly known for the hybrids of Paphs and Frags. QF Halia is a hybrid of Covacii, Longifolium, and Bessii. And this photo led me to their website, and yet another great opportunity for online shopping. John also shows us the Sophronitis, also known as Cattleya monticarii, a Brazilian species mounted on what looks like manzanita with some sphagnum tucked around it for extra moisture. The flower is very large for the plant size, and the petals are especially wide, full, and very striking. It's gorgeous. And this is John's Frag Fitz-Schomburg crossed with Sorcerer's Apprentice. This is an unregistered hybrid at this point. You can also see the Covacii and the Bessii in the background of this. The spots up in the upper part of the pouch are really striking. And Ron Norris shows us his Cattleya or Laleocattleya Mona Pink Hiromi, which received an AM in 2005. It has beautiful rose purple flares on the petals and sepals. This is about a 4-inch flower. Ron also shows us Cattleya Pink Splash, which is a lovely, but it's somewhat of a mystery. There are no photos of Pink Splash in Orchid Whiz or Blue Nanta, but it is listed as a cross of Cattleya intermedia by Sophrolalia Red Doll. And the color makes me think of bubble gum. I wonder if it smells like bubble gum too. And this is also Ron Norris's. This is in keeping with his pink and white theme here. He shows us this wonderful Peloric Phalcaeus polka bell. It's a Falbellina crossed with Pulcherima. And the lip Peloric petals, you can see these petals want to behave like the lip. There's even some yellow kind of shading that you would see in the lip. Give it a really unique form. KS is Kungster Orchids, so it's registered just recently and has not been awarded yet. Judy Carney shows us Cattleya Mini Purple, which is a primary hybrid of Pumila by Walkeriana. It's a lovely compact plant with flowers that usually come out in pairs like this. See little pairs of flowers. Judy's is mounted, but this is also a good windowsill plant in a pot as it doesn't get too large. Judy also shows us Cattleya maxima. You saw mine earlier. It was a semi-albiform. This is the tipo or typical form, color form from Ecuador, Peru, and Colombia. The yellow stripe that you see and the throat is a distinguishing characteristic of maxima. And the flowers are about 5 or 6 inches across. They're very long lasting and they're fragrant. So this is a real showstopper, Judy. Judy also shows us Sophronitis or Cattleya monticarii that we saw from John earlier. This is named for the mountain range in Brazil where they're found at about 5,000 feet. We saw one from John McCallan earlier. Judy's has 3 flowers and looks like a little bud forming back here. And they totally hide the miniature plant. Tanya Lamb shared a number of photos with us, so it was difficult to narrow it down to 3 or 4. This is Vanda Yipsumwa. It's a complex hybrid with beautifully
Lynn?
Yeah.
Sorry, my name doesn't have the B.
Oh, I'm so sorry. Oh my gosh. No, all the slides will have that because I copied it. I'll fix it before. No, I'll fix it, Tanya, before it goes online. Thank you. This is a complex hybrid with really beautifully presented flat, round flowers and with deep red tessellations. Remember tessellations from the giraffe, right? I'll fix that, Tanya. Sorry. And Tanya shows us her Cattleya maxima with a gorgeous ruffled lip and the characteristic yellow stripe down the center. That's probably a landing strip for whoever the pollinator is. And Vandicostylus lucneri bluebird. This is a plant a lot of us have in our collections. It's very rewarding because it loves to bloom at least twice a year. And Vandicostylus fuchs ocean spray, which is lucneri, the one we just saw, crossed with celestis. It's a lovely spray on a beautifully grown plant, Tanya. And Tanya shows us Masdevallia glandulosa with a nice close-up of the flower on the right. This is a cool grower from the cloud forest of Peru and Ecuador with lovely purple flowers and long yellow tails or caudae. Glenn Finch shows us Dendrobium virgineum, the virginal white dendrobium. The flowers are about 2.5 to 3 inches across. They're crystalline white with yellow to orange at the throat. You see little dots there. This is a species from Laos, Vietnam, Thailand. And like many of the dendrobiums, this needs a dry winter rest until the new growth start in the spring. And Glenn shows us Oncidium praetextum, also known as Gomessa praetexta. This is a Brazilian species which we don't often see in collections. It's related to Oncidium crispum, but according to J. Fell's Orchid Species Encyclopedia, praetextum is distinguished by the witch's nose on the lip. So I tried to zoom in on one of Glenn's flowers, but I can't quite see a witch's nose. Maybe there. Lovely flowers nonetheless. For Mary Garrettson, we see a Dendrobium canberriense, endemic to Thailand, in an area that is hot with high humidity. It's a miniature orchid with charming little flowers that are about 1/4 of an inch. These are photos by Ron Parsons. This fuzzy little beauty from Mary is Dendrobium hecuense, named for the Hekou province of Yunnan, China, and first described in 2011. J-Fel indicates that it grows on the underside of branches, so the flowers do not get water in them, and it grows at elevations of 1,000 to 2,000 meters. It's really a miniature sized cool growing epiphyte that is just darling. Mary also shows us a lovely dark form of Cattleya clandii, a favorite Brazilian species with a typical magenta purple ruffled lip. It's found in low elevations from up to about 1,200 meters, so it's an intermediate to warm grower. It's a bifoliate species, so somewhat temperamental to grow. It has a lovely spicy fragrance. Chani shows us this Cattleya, Cattleya Lulu Hot Pink by Cattleya Orchidom Brabant. It's an unregistered hybrid, and I just think the clear color is lovely. It gets all these spots from Cattleya clandii, which is in its family tree, which we just saw clandii from Mary Garrettson. This is Chani's Paf Grayi, a primary hybrid of Paf Godefroy by Pafnivium. They're both in the Brachypetalum section of the genus, and both are from Thailand. This is a great close-up shot of the interior of the spotted pouch and the white staminode with a yellow in the center. Chani also shows us an unregistered, recently made hybrid, Cycnoches Varzovixii by Cycnoches Kevin Clark, both with significant AOS awards. Cycnoches is in the Catacetinae tribe, so it's deciduous and requires a serious dry rest all winter. Chani says the natural horizontal spread from tip to tip is 13 centimeters, so that's about 5 inches. And the natural vertical spread is 11.5, so 4.5 inches. So with this parentage, no wonder it's a big beauty. Kay Klum shows us this beautiful Masdevallia, which was given to her by Anna Chai. The hybrid Masdevallia Red Shine was made by Tom Perliti, and he named the cultivar after his wife Sheila when it received the highest AOS award of first class certificate. This is Kay's beautifully grown Barbacella conioiana, a miniature, as we can see from the 3-inch ruler she's included here to give us the scale, and it's an epiphyte from Brazil. A little close-up of the flowers here. Kay also shows us a Platystellae stenostachya, a miniature epiphyte from Mexico through Central America, again with a 3-inch ruler to show us the scale. It has translucent yellow flowers that are about 1/4 of an inch in size. And another Platystellae from Kay, Platystellae misera, the name meaning poor or insignificant. Platystellae, the poor thing. How would you feel? And finally, this is Mary Nesbitt's Encyclia tampens. As many of you know, Mary owns California Orchids in Bolinas, and these are her 2 inquisitive 1-year-old kittens in training to become orchid judges. On the left, you see Monkey, and on the right, Dahlia. And I thought it would be fun each month to end show and tell with one Orchids with Pets picture. So please send me yours. And if I get more than one, editor's choice, or I'll save it for another month. So I welcome your feedback on this if you like the bigger, more pages with fewer orchids, and on the show and tell prezzo overall. Thank you.
Thank you, Lynn.
My pleasure.
Thank you, Lynn.
You want me to do Orchid Doctor?
Sure. Keep on going.
Okay. Let me click on down here. So the board has Received requests to include an Orchid Doctor or mini skill session in our virtual meetings. And Jeff asked me to go first. So on the left, after I watered this healthy looking catasetum, whoops, sorry, I went too far. After I watered this healthy looking catasetum last week, I took a peek at the underside of the leaves, and what did my wondering eyes did appear but spider mites. Yuck. So thin leaf plants like catasetums, lycastes, a lot of the terrestrials are very prone to spider mites. So I checked Mary Garrettson's book, Bay Area Guide to Orchids and Their Culture, and learned that the spider mites are microscopic arachnids, which feed on the juices of the leaf tissue. She writes, quote, "The black spots are likely sooty mold, which grows on the excrement of the mite infestation." Ugh, yuck. So spider mites are most active with warm temperatures and low humidity. And I know this plant—
Nothing like a little poop.
Yeah, little baby poops. So I know where it's located in my greenhouse, there's not a lot of airflow and the fans don't reach it well. So these bad boys have to go. So a little research showed that most insecticides do not work on spider mites because they're not insects, they are arachnids. So they have 8 teeny little legs. So you need a miticide. And I generally prefer an organic solution whenever possible. And products containing spinosad have been around for a long time. It's derived from the fermentation juices of a lowly soil bacterium called Saccopolyspora spinosa. So I Googled, does spinosad kill spider mites? And it says spinosad products can be used to directly kill spider mites on contact, can also be used when watering plants to systemically kill spider mites via the roots. Spinosad can also be effective at fighting caterpillars, thrips, and many other marijuana pests. Was more than I needed to know, but interesting. So Monterey, this brand, Monterey, is just one brand, which my local nursery carries. There are many other brands which contain spinosad. So
Lynn, just
yeah.
a point about spider mites. This is Mary, so everybody knows who's talking.
I know your voice.
They actually thrive when the humidity in your greenhouse goes down, especially in the summer. So one of the ways to discourage them is to keep your humidity higher if possible, because they just multiply like crazy when the humidity is lower, which obviously happens in the summer.
We had a couple of hot spells, so that's obviously what happened. So yeah. I drenched the leaves with spinosad solution 4 days ago. And I hope that means that these spider mites are dead, but the sooty mold did not disappear, so it needed to be washed off. And you can use products like Safer Soap to do that. But what I had on hand in the greenhouse was ivory dish soap and warm water. And of course, I was thrilled to later read that what I'm wiping off and have all over my fingers is the mold which grows on the excrement of spider mites. So hopefully I caught the infestation here before it did significant damage to the leaves of the catasetum, but the leaves are going to deciduate soon anyway. That's not a verb, it should be deciduate. But I learned my lesson to check frequently for spider mites and get them before they spread to neighboring plants. And that's it.
That's great. Thank you.
Welcome. So how do I give you back stop share? Here we go.
Yeah, Lynn, my dad was a pest control advisor for like 40 years, and he used to say the difference between spider mites and insects is you can put a insecticide out on a surface and insects drag their abdomen through it as they crawl. Spiders walk on their tiptoes.
Yeah.
So their body never touches the surface. So a normal insecticide doesn't work. So, and also that's why most systemics don't work, because spider mites are so small they can plug in anywhere, but things like scale tend to plug into the vascular system directly. And that's where the systemics usually work.
Good to know. So hopefully my spider mites on that plant are gone. I'll let you know.
If it tells to kill spider mites, it kills spider mites. Go for it.
All right. Any other questions, comments?
I have a few plants to show. I don't know if I'm the only one.
Keep on going. Go, Bill. You're on.
So quick and dirty. Host has disabled screen sharing.
Wait a minute. How about this? We'll make you co-host. How's that?
Let's see. Okay, well, okay, there we are. So this is, we're talking Sobralias before. This is Sobralia yapiensis by Xanthaluca. This doesn't do it justice. It is really yellow. This is Fimbriata by Sobralia P. I'm assuming it's Macrantha P because of the purple. It got a little bit of Fimbriata still in it, and it's about that big around. It's a tiny little plant. Well, tiny little flower, big plant, tiny flower. Embryi, Stanhopea embryi. I wish I could make this smell through the system. It is wonderful. It's the best Stanhopea scented one I've ever had. This one is, I think it's Tigrina, but the tag is lost somewhere in that giant plant above it there somewhere. And last but not least, Cattleya Irene Tarolang Kong. And it's one of those ones you were talking about, Circle of Life hybrids. That's one of the Circle of Life hybrids. And it's a little one, little plant, big flower. That's it.
Okay, thank you. And I was multitasking. Did the other Jeff Harris, did you get to his, or they came in too late? Lynn, I think they came in too late, right?
I didn't see them. I was probably having dinner. What time did they come in?
Very late.
Yep. So Jeff, I'll put them in next month if that's okay.
That's totally fine.
Okay. Thank you very much for sending them.
Okie doke. Let me share my screen. Where are we here? Okay. Let's see. How do I take back? Yeah, I'm trying to introduce him. Hold on one second. I just figure out what I did here. Zoom, 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 zoom. Share screen. There we go. All right, let's see. So, Jason, thank you for your patience. This is Jason Miller. I mentioned at the beginning that he's going to give a talk on conservation of the ram's head orchid. And I won't read everything. It's in this slide here, but basically new to working closely with native orchids. Before moving to Wisconsin, he worked as a program assistant in the rare plant program, and got interested in native plants. And he's been volunteering in this program. Introduced to a land manager in Door County, which I assume is in Wisconsin, and learned of the high diversity of orchids in Wisconsin and conservation effort that was ongoing. And he began working with Dr. Lisa Grubisha. Is that how you say her name? I'm not sure if I can. Grubisha. University of Wisconsin, Green Bay, and the North American Orchid Conservation Center. And he applied for funding for his research, and it was approved last year. And we asked him if he could come back and talk about his project. So the project, he'll tell us a lot more about it. And I will stop sharing and let you take over here. Hold on, let me find you. Okay. And try it now.
Well, first I want to say thank you guys for having me, and also for choosing to fund my research, but also just for having this opportunity available for anyone to be able to apply for funding. And I mean, it's critical for graduate students to learn how to write grants, and to be able to fund their research. So I do thank you guys all. So without further ado, let's start this up. There we go. So like just said, my name is Jason Miller. I am a master's candidate in the Environmental Science and Policy program over at the University of Wisconsin in Green Bay. So yes, Door County is in Wisconsin. If you look at a map of Wisconsin, Door County is the thumb that sticks out into Lake Michigan. So we have a really unique climate. We're surrounded by water. We have the Bay of Green Bay on one side and Lake Michigan on the other side. So orchids, yes, they do grow in Wisconsin. Most people I talk to are really surprised to hear that orchids grow here, because as you guys have shown in your presentation, a lot of things are tropical. So you think of Wisconsin, where we can easily get 100 inches of snow in the winter. To have orchids growing here is just amazing to me. Our state has 40 species, which is about the midway point for orchid diversity in North America. And actually in Door County, we have over half of the species here. Sorry if I keep looking away. I have two screens going because my printer decided to bite the dust, so I couldn't print off my presentation. So I'm going to talk a little bit about just the diversity that we have here in Wisconsin. So we have our evergreen orchids, or Licudieras. They're really known for their leaf patterns, which stay green all winter. They just have some intricate venation, and then they flower later in the summer. We also have microheterotrophic orchids, or the coral roots for those who know them, or Mycorrhizal. We have 4 species that are here in Wisconsin. On the left here, the little green one is Trifida, and then the other one is, I believe it's the striped coral root. They could depend primarily on the mycorrhizal fungi for all of their nutrition. They do photosynthesis a little bit, but not very much at all. One of my favorites actually here is the grass pinks. There's just something so fascinating to see the beach scene. They grow more in the dunes or sandier soils here, but you just see that little pop of pink through all the green vegetation, which I just find to be really fascinating, because otherwise you don't see them because they're so small. And then I'm going to get talking into the Cypripediums, which is where my research lies. We have the third most diverse population, or the third most diverse genre in Wisconsin. It's followed, the other two are Spiranthes and Panthera, which are still cool, but I find the lady slippers to be a little bit more show-stopping. So here on this slide, we have the pink moccasin lady slipper. I guess you can call this the showstopper of the lady slippers. It's the showy lady slipper, which, as you can see by the rule there, is probably that one there is probably about 2 feet tall. So it's just big and vibrant, and the deer love to eat them as well here. Where I have been doing my research is with the yellow lady slippers as well. I've been studying the genetic diversity of that one through another grant I received. They are very tricky, as you see in the pictures here. They look different because there are actually 2 varieties that grow within Wisconsin, and the biggest determining factor is the color of the sepals, which aren't always clear as day sometimes. They have a lot of variability to them. And then the research that was funded partially through the grant here is for the ramshead orchid. This one here is the smallest species in Wisconsin. It's only up to about 12 inches tall. So it's super small, and they have a ram's head shaped flower pouch, which given the common name makes sense. They do have a very restricted range. As you see by this USDA map, they are stretched throughout part of Canada and just the northeast portion of the United States. And wherever they're found, they're considered rare, endangered, or threatened. And here in Wisconsin, they are considered threatened. There was a population study done by the Forest Service a few years back, which estimated about 320 populations or so that are known to exist. I don't know how they consider populations. I think that's very vague based upon each state or province. So, but here in Wisconsin, its range is restricted more to the north. So there are 34 recorded occurrences from 15 counties in the state, but only 24 of those have been found within the past 20 years. So it's not looking great for our state here. And then these pink dots here are representative of populations that are either on private land or are no longer here. The ones that kind of are south of the bay there are the populations that are presumed extirpated. And the one that's the farthest south is actually Milwaukee. So there's probably no chance that that population still exists. Given that these plants are so small, the populations are super challenging to find. And of course, they don't grow along the beaten path. So you really have to bushwhack to even go find them. And then, as Jeff mentioned earlier, there is a rare plant monitoring program here in Wisconsin, but monitoring is not very regular for most of the species, but especially not for the super tiny orchids. So they may have been reported on at one point in time, but the data has not been collected upon again in many years. So a lot of what I'm doing is actually also scouting for refinding these populations. So it's been an adventure so far this summer. So I have 3 areas that I am interested in for this population, for the species. So I'm looking at the population genetics, which has been funded by the Orchid Society here. Also the microsite requirements, and also can't really study orchids without looking at the mycorrhizal symbionts. So for the population genetics, I'm really interested in the structure and the diversity of the populations and how the gene flow are moving throughout the state. So on this map here, the blue dots are populations that represent where I have collected leaf samples. Unfortunately, it's not looking so great. It's turning more into a Northeast Wisconsin study, but Door County is really a hotspot for orchids, so I can't say I'm very surprised by that. The red dots are actually populations where I have already searched and plants were not found, but I made soil collections, which I'll be talking about in just a little bit there. Another reason to study these genetics is they really give an insight into how a species can adapt to climate change or any other sorts of changes that might come its way. So as the climate warms, these populations are going to suffer because they actually require cool soils to grow and germinate in. So that's probably why some of these southern populations in the state have been lost over time. And then habitat loss and fragmentation is an issue for all species, but especially for the Cypripediums, they act as a deceit for pollination. So they don't offer a reward to any of the pollinators. So pollinators learn, and then as these populations become more fragmented and more suitable habitat is lost, it really can restrict how the gene flows throughout. And one other bit I was interested in for population genetics is the clonal diversity. So Cypripedium species especially are known to reproduce asexually, so they kind of will just create bigger and bigger clumps. So these 3 orchids here, there's a good chance that they may be connected underground, but of course I wouldn't know that unless I dug them up, which I wouldn't want to do. So what I would do is take leaf samples from each of these and then basically isolate DNA and then have the population structure and genetics run on that and see how similar they are to one another. What this can do is this can really help give insight into basically how similar populations are to themselves. The more similar they are, I will use the worse off they actually are, which is not a good way to say it, but it can kind of help land managers understand, like, okay, this population I have here is really like itself, and we need to do something about that. So that could possibly mean bringing in pollen from an outside source or from another population across the state, or even working with a researcher to germinate these in the lab and outplant new plants from a different population. The microsite requirements. So all plants need a specific thing to grow. Some are just really common, and then orchids just happen to be a lot more narrow about what they really want. So what I'm doing is, you can see in the picture here, I'm taking soil cores from each of the populations, and then I will run a suite of abiotic factors just to analyze them and see where do these populations stand, compare them to other populations throughout the state, and see how they compare to one another. How I plan to do that is, this is just a hypothetical population here. So the blue circle is what I'm considering the population, and then that green band around it is outside. So what I want to do is actually take soil samples from within the population and outside the population and see how they compare. So you would think orchid population would kind of just expand over time, but is there something stopping it from expanding? Is the soil outside of this population not suitable, or is it just, the orchid's just not taken over that area yet? So what I plan to do is, depending upon the size of the population, take 3 to 5 samples from each area and then run these analysis, so like pH, ammonia, nitrates, nitrites, soil moisture, things that could be easily run by land managers. They really wanted to search out from some new areas. And then what I plan to do after I get all of this data back, compare all the populations without any requirements. So just every population I collect soils from, just to see how they work or how they compare to one another. But I also want to compare sites with similar soil types. So this species actually grows in a variety of habitats. It's been identified as 3 different types. So it can grow in some sandy soils, but then it also has been found in soils that are a lot more mucky. So I wouldn't really want to compare those two, because it's like comparing apples to oranges. They're very different from one another. So I would want to break those sites apart and then compare like all the sandy soil sites to each other, than all of the sites that have more of a mucky soil. And how I'll do that is I'll use the USGS soil types just to kind of determine how they lay out to each other. And then for the mycorrhizal symbionts, the map here, the blue dots here show where roots have been collected. I really limit where I collect roots from to suitably sized populations. So I've come across populations that are only 5, 6, 7 plants. Those would be ones I don't want to collect from, just because they're already small in size as it is. I would not want to jeopardize the success or the future of that population. So I would opt not to collect from those. So this has, even though my population size, my sample size for roots would be small, it can still really give a lot of insight. So the mycorrhizal symbionts, it's not specific just to orchids. It happens in many different species, and typically it goes both ways. So the fungus will usually get something from the plant, and then the plant gets something from the fungus. But that's not the case with the orchids. So the orchids basically just take everything they need to germinate, and then also help with their dormancy when they go into dormancy for extended periods of time. So how I'm going to do the identification of mycorrhizal symbionts. So in this picture here, I've actually highlighted roots of a Cypripedium species. So what I'll do is, a couple of centimeters from the base of the plant, I will slowly excavate out the soil just to expose the roots. And then I follow that back up to the plant just to make sure that the root I have is actually going to the plant that I want it to. And then take that back to the lab and process it following protocols that have been developed by Dr. Larry Zettler and by the Smithsonian. They have different ways, so I like to do both of them just to see if one is more successful. And then using the same soil analysis, I wanted to also assess how the chemistry of these populations influences fungal associations. So this here is a picture of actually peloton growth. So what I do is I come back, go into the laminar hood, and then I just isolate underneath the stereoscope and grab out pelotons, or what I think are pelotons. They're very small and not always super easily identified. But then I put them onto petri dishes with gel media, and I basically just watch them until I see growth happening. This here is a strong contender for peloton growth, or one of the yellow lady slippers. Growing that one out now still, and we'll hopefully be having some answers for that one very shortly. There's been a little work actually done with mycorrhizal fungi for the species. A lot has been done for Cypripedium, for other Cypripedium species, but the little bit of research that has been done shows that this probably associates with either the Russula genus or with Tulasnella, which is comparable to the other species of Cypripedium. And then one other bit I want to do, so orchids require the mycorrhizal fungi for germination, as they have microscopic dust seeds, so they have no endosperm, so they need all the nutrients from the fungus. So what I want to do is actually quantify the amount of mycorrhizal fungi from the soil around plants. So in this picture here, the yellow dot represents the base of a plant. So what I did from there is I would just take a tablespoon or two of the soil, and then in the 4 cardinal directions from the base of that plant, at 15 centimeters and 30 centimeters, I will take a soil core sample and isolate DNA from each of these samples to quantify the amount of mycorrhizal fungi. I would just hypothesize that as you move away from the base of the plant, the mycorrhizal fungi abundance will actually lessen. But that's not always the case, but I will find that out. And also what I plan to do is any of the soil samples that were taken from outside of the population, so that green ring there, I also would want to isolate DNA from there and quantify the abundance to see if that may be a limiting factor to the population's expansion. So a lot of studies have isolated orchid mycorrhizal fungi from the roots, but very few have actually identified, have examined how location or habitat influence the fungal assemblage. So that's another issue I want to go over. There has been some evidence that shows differing populations across the range will actually have different fungal associations. So that's one thing I'm hoping to be able to answer from this part of my research as well. And then my next steps, so this past summer, I've actually started my research. I applied for this grant in December, so definitely not in the growing season for any plants here in Wisconsin. So this past summer, I spent time collecting root samples, soil samples, leaf samples from populations. I did start processing the root samples and have them growing on media in the fridge, and I will continue to work with those. I will actually make slides. So these pictures here, Tulasnella have very monoloid cell structures, so circular cell structures to have a rough estimate. That's a good characteristic of that. So I will actually make slides of all of my cultures I have just to see if they have this structure or they don't. If Fusarium is another common mycorrhizal or common fungi that can grow in the soil, and those also have a very distinct banana shape to them. So if I see a lot of culture of that in there, I would more than likely just dispose the sample, because it's not worth the time to have that sequenced out and just find out it's Fusarium when all I see in a slide is the Fusarium cells. Then I'll continue to work with isolating the DNA from all my leaf samples and have that whole process be done, and then start processing the soil samples, start drying them. They're currently sitting in the freezer. I'll dry them out and then send them off to the lab for analysis to have that done. And then I did not get to all the sites last summer. This past summer, it did not work out for my schedule. So I plan to revisit some sites that were not sampled. Those would be the ones represented in the red dots. And then the orangish dots, I want to revisit these sites. I got to them a little late in the summer, so the plants were not in prime flowering season. So I want to revisit them because the data that I have from the DNR here in Wisconsin indicates that there's a strong potential that these plants could still exist. So I want to just go back to these populations and search again just to see if I can find them, because that would help really answer some more questions of how the populations are doing across the state. And that is kind of an overview of what I am doing. It's not a lot yet because, like I said, it kind of was influenced by the weather, so hopefully things will be becoming more positive.
Thank you. Any questions? Someone asked what a Peloton was.
Oh, it is not a bike. It definitely sounds like a bike. I guess I can stop sharing my screen because no one needs to see that presentation. So it is a fungal coil that kind of just lives inside the root, and what that does is it gets infected by the fungus, and that's how the plants germinate.
Cool. Other questions?
Hey, Jason, thank you for the presentation. I grew up in Minnesota, and you know, my parents you know, gone on vacation to Door County and so on. And they have a place in northeastern Minnesota where they have a form of lady slipper. I'm not sure what species it is. I don't think it's showy, but they definitely are pink, not the yellow kind. But I guess this, I don't know. Let me see if I can think of a question about it. But yeah, what do you know, what sort of time range are you seeing on the flowering? Because I haven't managed to be there when they're flowering. Theirs seem to flower sometime in June, but what time frame?
I think it varies. I think for Minnesota, that would make a good sense for the ramshead to be flowering in June. Here in Door County, it's, I would say, kind of like late May to early June. It's hard because they don't last very long. Some studies have shown they only last for about a week after flowering. So it is a short window. And since they are so small, if you don't know where to go to find them, it's kind of like a scavenger hunt, finding a needle in a haystack.
Jeff, you had a question about whether the fungi are responsible mostly for germination or ongoing nutrition for mature plants?
Well, definitely they're definitely responsible for germination. For the ongoing nutrition for mature plants, there's not a good consensus for that, honestly. There has been some research done that shows after the loss of the mycorrhizal fungi, the population will be lost within 2 years. So it's presumed that there is something still going on, especially when the plants are dormant. But there's not a hard rule for that, unfortunately, which would be nice if there was some better data for that.
Yeah, I was kind of curious to see if anybody's done something like, I mean, you can give you know, radioactive nitrogen sources and things like that, and see if, you know, the fungi are harvesting that and bringing it into the plants. So I mean, obviously the fungi are, you know, throughout orchids are known for being responsible for the germination, but that's the question is, if you go like the Mycorrhizal, where it's clearly you know, a mycotic interaction that allows them to survive, where along that continuum do these guys fall? If that's a critical factor just in their germination, or if that's a critical factor in their ongoing sort of survival.
Yeah, no, that's a good point. Yeah, I would say it's not critical, but in times of stress, it's really important. Like times of drought or just a bad year for orchids in general, when they don't decide to grow above ground vegetation, then I think it's critical. But yeah, I haven't seen anything saying, anyone studying it in a fashion like you've suggested, or even being able to give concise information saying, yes, it is continuing nutrition for the mature plants.
Cool, thanks.
Okie doke. Any more questions? Joe has prepared the raffle behind me, so if there's no more questions, I want to thank Jason again. Thank you for all you're doing and for explaining it to us, and keep up the good work.
And once I have some results, I will definitely give you guys a report for this so you guys can see what's going on with it. So
We'll look forward to that. Thank you.
Yeah, glad we could help.
Yes, thank you guys.
Jeff, while Joe is pulling names, could I make one quick announcement? This is Lynn.
Of course.
About judging. So the PAC Central Judging Center is not able to judge because, as Jeff said, our meeting facility is not available. But California Sierra Nevada Judging Center has found an outdoor location at Dueling Dogs Brewing Company in Lincoln under a tent. So they've done one outdoor judging there. It was successful. We had 23 entries and awarded 13. So the next outdoor judging is Sunday, September 13th at 9 a.m. in Lincoln, California at Dueling Dogs Brewing Company. This is at 3030 Barrett Park Lane in Lincoln. This is near Allen's Greenhouse. You can look it up online. So plant entry starts at 8:30, and this is before the brewery opens, so there's plenty of room to spread out. And by the time we finish, the brewery is open, so you can have beer and a food truck comes. So we welcome you to bring orchids for judging. Thanks.
Thank you. 8:30 in the morning, I assume, right? Yeah. Any other announcements?
That's kind of early for beer.
You think? In these times, Mary?
All right.
Jeff, can we put that judging information on our website, or will it be on the newsletter?
Yes, I wrote it down. So.
Well, since it's only 2 weeks away, it probably won't be able to get to next newsletter, right?
True.
So maybe website will do, or
Yeah.
email.
Maybe put it on the Facebook. Can you do that? Put it on the Facebook page?
Sure. Actually, Lynn, can you send me an email?
Yes, I will. Right?
It'll remind me to do
I
it.
will.
I forget things. There's too many things.
I know. Here we go.
All right. Are we ready? Okay. So the first winner of the raffle tonight is Corey Majewski.
Yay.
I saw you. Second one, Lillian. Lillian's a new member, right? I think Lillian, are you here? You're here. If you come off mute and say hello.
Hi, I'm thrilled I got to win the raffle.
Yeah. Welcome and congrats.
Thank you.
Let's see. Lynn, you're a winner.
Yay! Thank you.
Allie Fishman. Are you? I think I saw her.
Here. Hi.
Hi. Congratulations.
Thanks.
Let's see. Christine. Oh, this is Mickey. Okay. Mickey, you're a winner.
Thank you. Lucky me.
Okay. Angelique Fry. Are you still? I think I saw you on. Is she still on? I'll follow up with her. Angelique, are you on still? If you can unmute. All right. Check. Paula Shu, I think you told me you were on, right?
Yep, I'm here.
You're a winner. Jenny Lum. Jenny, are you on? Yes.
The video go.
Yeah,
Hello.
I'm on. Thank you very much.
Okay. 1, 2, 3, 4, 5. Wait. I'm making sure I don't overdraw. Okay, 2 more. Heidi Arno. And Gia Lee. Gia's on. I saw her.
Hi.
Hi. Okay, I think that's it. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So I will follow up. I'll try to get to it tomorrow. And if not, by the next day. But I'll send the information. You've won a $20 gift certificate, good to shop online at the Tiny Jungle orchid greenhouse, and you can buy it online, and we cover shipping. So there's some instructions on how to do that. So I'll send you all that with the code that you use to claim your prize. So thank you for attending. What else? Reminder to look over the next 2 weeks and send us lists if you're want to sell plants. And then also we will send out announcements about when the after October 1st, when the sale goes live, and we'll be having our meeting around that time too. So stay in touch, stay safe, and good to see everyone.
Good job, Lynn.
Okay, yes. Thank you,
Yes,
Lynn.
that was excellent, Lynn.
Good night, all.
We
Good
don't
job,
need Ron
Lynn.
Parsons anymore at show
Yes,
and
we
tell.
do. We want Ron. We want
We
Ron.
miss him too, yes.
Actually, I
That's a
could use
compliment.
people to do orchid doctors or skill sessions too. So let me know if you want to help out.
Okay.
All right.
Thank you, Jeff. Thank you, Lynn.
Bye. Hi,
Good
Valerie.
night.
Oh,
See you
good
guys.
meeting.
See ya.
Thank you.
Lisa.
Thanks, everyone.
A fun meeting. Good one.